So very good afternoon. So we will wait for some time and let others join, and then we will start today's session. I hope my voice is audible and the video is visible. So once again, very good evening and welcome to the week 2 live session of NPTEL course Medicinal Chemistry. I hope my voice is audible and you can see the presentation. Please let me know in the chat box if my voice is audible to you, and also if you can see the presentation clearly. I hope my voice is audible to you. Please let me know in chat box. Hello. So is my voice is audible? Okay, thank you so much. And the PPT is also visible, right? We will wait till it is 6 p.m. and then we will start the session. So last day what we did, we discussed about the week 1 and week 2 course content, the topics we have learned in week 1 and week 2. We discussed those topics and we discussed 20 questions regarding those topics. So I hope the video and the presentation is has been made available to you by NPTEL. You can find it on the SWAM website. Okay, if you go on to the SWAM portal, you will find there the problem solving session video and also the presentation. Today it is our week 2 live session where we will discuss the week 3 topics. Meanwhile, if you have any problem while listening to my voice or seeing the PPT, please let me know. So I think I can start with the introductions. So very good evening to all. My name is Mousumi Deb and I am the PMRF research scholar or the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your NPTEL TA for the problem solving sessions of the course Medicinal Chemistry. So today it is our week 2 live session. So in previous session we had done the week 1 and week 2 course content together in a single session where we discussed 20 questions regarding those. So today it is a week 2 live session and right now the week 3 of the course is ongoing. So today what we will discuss is the week 3 course content. The topics which are included in the week 3, we will discuss those topics in today's session. So basically we will discuss 20 MCQ questions just like the last day. We will discuss the 20 MCQ questions and for each question I want all of you to try to answer the questions. So I hope my voice is audible to you and the presentation is visible. If anybody has any problem while listening to my voice or seeing the PPT, please let me know in the chat box. So for the smooth running of the session, and also I am recording the session, that's why I have muted you all and also I have disabled the video option. But if you have anything you want to say or you want to communicate with me, the chat box is always open and I am monitoring the chat box. So if you have anything you want to say, you can write it on the chat box. Okay, I will check it, and if you have any query, any doubt, any question, everything you can write it on the chat box. And also I always share my email ID at the end of the session. So if you have any problem, any doubt regarding any course content or anything, you can drop an email to me. Okay. So before starting, I will just clarify few things. So I hope everything you can see the PPT. If not, you can just write it on the chat box. So as I said, today it is a week 2 live session. So this is a problem solving session and which we will continue for next few weeks. For this is a 12 week course, so we will have 12 problem solving sessions. In addition, we have one summary problem solving sessions at the end. Okay, so every problem solving session it will be of 2 hours. Okay, we will having this kind of problem solving session every Tuesday. Okay, so we will have this kind of problem solving session every Tuesday from 6 PM to 8 PM and these sessions will be live on Google Meet platform. So if any network disruption happens from my side, so please do not leave the class immediately. Okay, wait for a few minutes, I will join as soon as possible. And if you have any network disruption and anything happened, so don't worry, because I am recording the session and I will be uploading the session, and the link will be shared to you on the SWAYAM web portal. So you can check the recorded class from there if you have any internet problem. But if not, I want all of you to join as much as you can on in the live sessions, because it is the only way we can interact with each other. And so if you have any doubt, any queries, as I have said, for a smooth running of the session and also for recording purpose, I have just muted you all. But if you have anything you want to share, you want to tell me any doubt, any queries, anything, you can just write it in the chat box. The chat box, as you can see, is open and I am monitoring the chat box always. So if you have anything you want to tell me, you can just write it on the chat box. Okay, at the end or in the in between I will try to answer your doubt, your query. And if still any doubt persists after class, you can drop an email to me. I will share my email ID at the end of the session like last day. So you can just drop an email to me regarding your query, your doubt. We can discuss it in the next class. Okay, and this presentation and class recording, as I have mentioned, it will be shared with you on the SWAYAM portal. So you can just log in to the SWAYAM portal and you can find it there. So now I hope my voice is clear enough and you can see the presentation, right? Just anybody can write it in the chat box if the presentation is visible properly, then we can start the session. You can see the PowerPoint presentation. Yeah, thank you so much. So we will move forward. So as I have mentioned, today it is the week 2 live session, and in this week 2 live session we will discuss week 3 course content. So what are in the week 3 course content? First we will start with nucleic acids. So nucleic acids, like we all know, the very common nucleic acids or the only nucleic acids which are we are very familiar with are DNA and RNA, right? So we will discuss the DNA and RNA, the basics and the structural functions, and then we will go to the RNA and protein synthesis. We will have nucleic acid and basics of molecular biology a little bit. Then we will discuss enzymes as drug targets, enzyme kinetics and inhibition, and various modes of inhibition today we will discuss. Today I will give you some mathematical problems too in the during the class. So I will show you the answer how to solve those things, but before that I want all of you to try to answer these things, okay, because these type of question and everything will help you to submit or to solve your weekly assignments, okay. So try to concentrate and let me know if you have any doubt or any query during the class. So we will now start, and when more students will join, I will just let them in, okay. So let us go to the first question of the session. So the first question is the very basic one. So what is the correct composition of nucleosides? Okay, the correct or the options are: number A, base plus phosphate; number B, base plus sugar; number C, sugar plus phosphate; and number D, base plus sugar plus phosphate. So what is the correct composition of nucleosides? All of you try to answer the question and let me know which of one of the following options are correct: A, B, C or D in the chat box. Okay, I am getting few answers. Okay, others also try. I want all of you, see, in these questions or answering these questions won't give you any marks, or it won't deduct any marks if you have given wrong answer. So I want all of you to try to answer the questions. What is the correct composition of nucleosides? Base plus phosphate, base plus sugar, sugar plus phosphate, or base plus sugar plus phosphate? Somebody wants to join. Okay, so I have few answers. Most of the answers looking like it is number B, and I have option D also as answer. Okay, now I will show you the answer and we will discuss about it. It's good that most of you are giving the answers. That is required for this kind of interaction sessions. So the correct answer for problem number 1 is, okay, somebody wants to join, let them in, then we can discuss it. Okay, so for problem number 1, which was correct composition of nucleosides is, so the answer will be number B, base plus sugar. So most of you have given the correct answer. So now let us discuss. So basically nucleosides, nucleotide and nucleoside, there are two terms most often the students get confused. Okay, what are nucleotide and what is nucleoside? So see, basically in any your nucleic acid there are 3 common parts. Okay, so the 3 basic parts. One is a nitrogenous base, then it will be one is a pentose sugar. 1 second. Okay. So now what are the common parts of the nucleosides? So as you know, nucleosides or nucleotides, these two are very common term, but often the students get confused which part is nucleoside and which part is nucleotide. Like in the answers which you have given, there were 2 most frequent answer, right? One number B, which is base plus sugar, and other is number D, which says base plus sugar plus phosphate. So most of the student get confused which one is the nucleoside and which one is the nucleotide. So let me tell you, nucleoside is one with the only base and your base and your sugar. So with base and sugar, okay, it is called the nucleoside. Okay, and if we have one phosphate unit along with this sugar part, okay, then it is called the nucleoside monophosphate. Okay, but if we have 2 phosphate units together, it is called nucleoside diphosphate, and if you have 3 phosphates, it is called nucleoside triphosphate. Okay, and if for this portion with this base, sugar and phosphate together, it is called nucleotide. Now, based on this base is a nitrogenous heterocycle. So, we all know in nucleic acid, DNA and RNA we have basically 5 type of bases, right? Adenine, guanine, which are the purine type of bases. Purine means this ring is called the purine ring. One 6-member ring is fused with one 5-member heterocyclic ring. So, the numbering goes like this. This nitrogen which is next to this NH2 group in adenine, it is the number 1. So, goes like 1, 2, 3, 4, 5, 6. Now, we have 7, 8 and 9. This the nitrogen which is on the 9 position, from this position we have this particular sugar group. So, this is a very common type of question which is asked in many examinations, like from which nitrogen the pentose sugar is connected in nucleic acids or in nucleotides. So, you have to remember if this is the 9th nitrogen or N9, okay, from which the particular pentose sugar is attached, this nitrogen, 9th nitrogen. It is same for this guanine too. So, for adenine and guanine it is the 9th nitrogen, and for cytosine, uracil and thymine, which are basically the pyrimidine kind of base. Okay, this 6-member nitrogenous ring which have nitrogen in 1 and 3 position is called pyrimidine, and these bases are called pyrimidine containing pyrimidine base like cytosine, uracil and thymine. Okay, and so for this particular pyrimidine, the pentose sugar is attached with the the nitrogen number 1 with N1. So, remember for purine it is the N9 and for pyrimidine it is the N1. Okay, and then this for pentose sugar it is the first carbon from which the base is connected, and it is the your 1, 2, 3, and this is the 4th. Okay, this is the 4th carbon from which the particular hydroxyl group is there attached, and this is the 5 prime, okay, which is the CH, and there the phosphate we have. Okay, and this is the basic units of the particular nucleotide, and based on this 2 prime position, if we have hydroxyl in this 2 prime position, it is called the ribose sugar, and the ribose sugar is present in the RNA, and if we have hydrogen in the 2 prime position, it is called the deoxyribose sugar, which is present in DNA. So, this is very basic things about the nucleic acids, basic unit about the nucleic acid, but there are so many informations which you have to remember. Now, for the structural portions, it is very good if you can remember the structures of this purine and pyrimidine bases, because in many times there are questions regarding the structures also. So, remembering the structures will also help you for solving the same type of question. This is looking like very difficult, but if you practice for few times, you can just easily remember the structures of the purine and pyrimidine bases of the particular nucleosides. So, what we learned in this from this question, the basic unit of nucleic acid based on the nucleoside, which is only base and pentose sugar, and the nucleotide has base, sugar and phosphate together, right. The purine bases are adenine and guanine, which has this fused ring structure where one 6-member ring is joined with a 5-member ring, and in the pyrimidine we have a single ring with have a nitrogen 1 and 3 positions. Okay. So, this was the problem number 1. So, now I will move toward the next problem. So, problem number 2, it is a mathematical question. It is a little bit of math in this question. So, it is said that in a particular human DNA, it has been observed that adenine is present in 27%. So, what will be percentage of cytosine in the DNA sample? The options are number A 27%, option B 46%, option C 73% and option D 23%. Read the question carefully, try to solve it and let me know in the chat box which one will be the correct answer. I will repeat, in a particular human DNA, it has been observed that adenine is present in 27%. So, what will be the percentage of cytosine in that DNA sample? Your options are number A 27%, number B 46%, number C 73% and number D 23%. Take some time, but I want all of you to answer this question in the chat box. In a particular human DNA, it has been observed that adenine is present with 27%. What is the percentage of cytosine? 27, 46, 73, 23. Let me check the chat box now. Okay, so I have few answers. Number A, number A, number D, 23%, number A, number C, number A, number A. So I have many answers, I think. So I am saying it is number A, number D, number 23, number C, A. So I have A, C, D, 3 answers I have, right? Okay, let me check which one will be the correct answer, and then let us solve this question. So the correct answer will be number D, 23%. Okay, so why it is so? So basically in particular human DNA, what is the rule of base pairing? So for DNA, cytosine, it forms hydrogen bonding with guanine, and adenine, it forms hydrogen bonding with thymine. So what is the number of hydrogen bonding from between these base pairs? In case of cytosine and guanine, the number of hydrogen bonding is 3. Okay, and in case of adenine and thymine, in case of DNA, the number of hydrogen bonding between adenine and thymine is 2. And for RNA, cytosine is bonded with guanine again, so there is no change in this case. There are 3 hydrogen bonding again, and for adenine and uracil there are 2 hydrogen bonding. In RNA we have uracil, and in DNA we have thymine, which is bonded with adenine. So now read carefully the question. So it's what it says. It says that we have adenine present with 27%. So adenine, it makes hydrogen bond with thymine in case of DNA, because it is DNA is mentioned here. Since 27% of adenine is present, the percentage of thymine will also be 27%, because adenine is bonding with thymine. So the percentage with which the adenine is present will be same for the thymine also. Thus the remaining 2 bases, cytosine and guanine, will be consist of total 100 minus 27 plus 27, means your 46%. Okay. Now cytosine and guanine, they make hydrogen bonding with each other, hence they are present in equal amount. So the percentage of cytosine, it will be 46 divided by 2, which is 23%. So now I think it is clear to you. You look at the solve very carefully, and if you have any doubt, if you don't understand how it is done, please let me know right now. I will clarify your doubt and then we will move forward. Anybody has any doubt regarding this question? Adenine, it makes hydrogen bonds with thymine. Since if 27% of adenine is present, the percentage of thymine will be same, 27%. So now the remaining is your guanine and cytosine, which will be present 100 minus 27 plus 27, means 46%. And since these are equal in amount, so we will have the cytosine, percentage of cytosine, 46 divided by 2, which is 23. Since most of you have written it as A, so now is it clear? Whoever has done the mistake, it is fine, but I want to know if it is clear right now, it is clear or not. I will repeat. If it is not clear, if anybody has any doubt, we can discuss it more. So is it clear to you, or do I need to repeat it? Okay, thank you for your response. So since it is clear, so now just remember this base pairing rule, and then if any in this type of question you can solve easily if you remember this base pairing rule. Okay. So now moving forward, I will move forward to the next question. So problem number 3, it says that the structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? Number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar, a phosphate group. If somebody wants to join, let me add them. Okay, so problem number 3, I will just repeat it. It says that which of the following statement is correct regarding the structure of DNA. Number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar and a phosphate group. Number C, the double helix of the DNA is stabilized by interaction between adjacent ribose sugars. And number D, each DNA sequence folds into a unique and complex 3D structure. So, which of the following statement regarding DNA is correct? A, B, C or D? Which one the following options is correct? Read the options very carefully and then let me know in the chat box. Again, I want all of you to participate and respond. Which of the following statement regarding the structure of DNA is correct? Number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar and a phosphate group. Number C, the double helix of DNA is stabilized by interactions between adjacent ribose sugars. And number D, each DNA sequence folds into a unique and complex 3D structure. Let me check the chat box now. Okay, so I have mostly number B. Okay, let me check. So, the correct answer will be yes, it is number B. DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar and a phosphate group. If somebody wants to join. Okay. Now, let us okay. Now, let us discuss. So, as I have mentioned in the first question itself, when I was discussing about the nucleosides. So, the chemical structure of DNA. So, the sugar phosphate backbone. Okay. So, we have a backbone of sugar and phosphate in DNA. So, DNA is a polymer made up of units called nucleotides. The nucleotides are made of 3 different components, a sugar group, a phosphate group and a base, and the 4 different bases are there: adenine, thymine, guanine and cytosine. So, this is the structure of adenine, this is the thymine, this is the guanine and this is the cytosine. So, what holds the DNA strands together? DNA strands are held up together by hydrogen bonds between the bases on adjacent strand. Adenine always pairs with thymine, while guanine always pairs with cytosine. Adenine is paired with uracil in RNA. So, in DNA, adenine is bonded with thymine, we have 2 hydrogen bond, guanine is with cytosine and we have 3 hydrogen bonds. From DNA to proteins, the bases on a single strand of DNA act as a code. The letter from the 3 letter codons which code for amino acid, the building blocks of the proteins. So, we have DNA which gives after transcription it gives the RNA, and the RNA forms the protein from by the process translation. An enzyme RNA polymerase transcripts DNA into mRNA or messenger RNA, and it splits apart 2 strands to form a double helix, and then it reads the strand and copies the sequence of nucleosides. The only difference between RNA and original DNA is that thymine is replaced by uracil. Okay, and we will discuss this all things later also. So, the multicellular organism, the mRNA carries genetic code and the cell nucleus to the cytoplasm, and here the protein synthesis takes place. This translation is the process turning the mRNA's code to protein. Molecules called the ribosomes carry the process and building up the proteins for amino acids code for. So, this all protein synthesis and everything we will discuss later on also. So, this was from the structure regarding to the structure of DNA, and I think most of you have given the correct answer. So, now let us move forward. So, problem number 4, it says that in prokaryotes the first amino acids in polypeptide chain is number A methionine, number B N-methyl methionine, number C N-formyl methionine, and number D all of these. So, which the first amino acid in the polypeptide chain? Which one is the first amino acid in the polypeptide chain? Somebody wants to join. So, I have few answers. So, what are the answers? Yeah. So, number C, number C, number C, and I have few number C, few number A. Okay, let me check which one of this is correct. So, for problem number 4, the correct answer will be very good, number C, N-formyl methionine. So, in prokaryotes the first amino acids in the polypeptide chain is N-formyl methionine. So, this is the structure of N-formyl methionine. Okay. So, we all know what is the structure of methionine. For methionine there is only NH2 here. Okay. But in case of prokaryotes we have N-formyl methionine, and which has a formyl group in this NH with this amine we have a formyl group here. Okay. So, this is called the N-formyl methionine, and it is the first amino acids in the polypeptide chain. So, initiator tRNA, methionine is the first amino acid incorporated into the protein chain in both prokaryotes, which in case of prokaryotes it is modified to N-formyl methionine, and for in the eukaryotes the methionine is the first amino acid which is coded. The initiator tRNA are the special tRNAs that recognizing AUG or GUG for start codon for prokaryotes and eukaryotes. Initiator tRNA is different from the one that insert internal methionine residues. N-formyl methionine is the first amino acid to be incorporated into the polypeptide chain in the prokaryotes. So, this is the promethionine, this is the N-formyl methionine. After synthesis of polypeptide, the formyl group is removed by a deformylase. Often the N-terminal methionine is also removed by an aminopeptidase as well as one or two additional amino acids. So, remember in case of normal eukaryotes methionine is the first amino acids which is incorporated, and for the prokaryotes it is the N-formyl methionine which is incorporated. Okay. The codon which recognizes the methionine is AUG, and the codon for recognizing the N-formyl methionine is the GUG, which is the start codon for the prokaryotes. Okay. So, now moving forward. Problem number 5, it says that the complementary strand for primary single stranded DNA is formed by options number A DNA ligase, number B DNA primer, number C DNA polymerase, and number D DNA helicase. So, which one is the complementary strand of the primary single stranded DNA is formed by? DNA ligase, DNA primer, DNA polymerase, or DNA helicase? Which is the correct option? Which enzyme forms the complementary strand of the primary single stranded DNA? All of you try to answer the question in the chat box. The complementary strand of the primary single stranded DNA is formed by DNA ligase, DNA primer, DNA polymerase, DNA helicase. Let me check if there is any answer and then we can discuss it. Okay, I have answers like number C, number DNA polymerase. Okay, let us check and let us discuss. So, the complementary strand of the primary single stranded DNA is formed by, yes, it is the DNA polymerase. So, what is DNA polymerase? So, DNA polymerase is the very important. So, in case of this DNA replication, many enzymes are very important. Okay. So, there is a DNA polymerase enzyme which forms the double stranded complementary strand of in the leading strand. Okay. And we have the DNA polymerase, DNA the helicase, we have other few enzymes which I will tell you in the course of this during this course. So, there are many enzymes which are important. Okay, DNA ligase, DNA primer, DNA polymerase, DNA helicase. So, there are many enzymes which are important during the DNA replication. The most important one is the DNA polymerase. So, there are two strands. So, when there is a double stranded DNA, first there is a single strand formation from the double stranded DNA. Okay, and when there is a unwinding of this particular DNA, this DNA. So, after the unwinding we have two strands. The first one strand, it is called the leading strand, and another strand, it is called the lagging strand. Okay, and because we have two strands, one strand goes from 5 prime to 3 prime, and other strand which goes from the 3 prime to 5 prime. Okay, and this DNA polymerase, it forms the particular strand in a particular direction also. So, first as you can see, this DNA, there are few free nucleotides, and the DNA polymerase takes the few nucleotides and joins together with a leading strand and with the lagging strand also. And then we have this helicase enzyme, and this helicase enzyme also helps while unwinding this particular double stranded DNA, and we have this portion is called the replication fork. Now, in which direction it works on, everything we will discuss later. So, remember, the enzyme which is important and which forms the complementary strand is known as the DNA polymerase enzyme. So, now problem number 6. So, it is a mathematical question. Okay, and we have discussed about the Michaelis-Menten kinetics in last week. We have discussed few things about this. So, this question is related to that particular equation. So, it asks that if the ratio, what is the ratio of V0 by Vmax when the S or the substrate concentration equal to 9 km. So, if the substrate concentration equal to 9 km, what is the ratio of V0 by Vmax? Your options are number A 9 by 10, number B 10 by 9, number C 9 by 8, and number D 1 by 9. What is the ratio of V0 by Vmax when the substrate concentration is 9 km? Option A 9 by 10, option B 10 by 9, option C 9 by 8, and option D 1 by 9. This is from the Michaelis-Menten equation. You have to use that equation to solve this question. I think we have discussed that equation in the last class, and we will again discuss this kind of enzyme kinetics mathematical equations today's class also. So, this is very basic question. Just remember, try to remember what is the particular Michaelis-Menten equation, and then if you put the values you will get the answer easily. Now, all of you try to answer this question, all of you please try to solve the mathematical question and let me know what is the answer. What is the ratio of V0 by Vmax when the substrate concentration equal to 9 km? All of you try to solve this question. What is the ratio of V0 by Vmax when S equal to 9 km? Let me check the chat box now. Okay, I have different answer for this question. Number A, number C, number D. Okay, for this question I have A, B, C, D, 4 answers all together in one question. So now let us check which one of the following is the correct one, and then we will see how to solve this kind of problem. So the correct answer will be number A, 9 by 10. So this is the Michaelis-Menten equation, right? So we have this equation V0 equal to the Vmax into S, which is the substrate concentration, divided by Km plus S. Okay, so here the V0 is the initial velocity in the mole per time. Okay, S equal to your substrate concentration in molar unit. Vmax is the maximum velocity, and Km, which is the substrate concentration at half Vmax. Okay, and it is also known as your Michaelis-Menten equation. So now if you rearrange this equation a little bit, and then if you put this particular value, you will have this particular and the answer, the correct answer for this kind of question. Okay, so now let us do it. So this is the initial equation, which is V0 equal to Vmax into S divided by Km plus S. Now if you take this Vmax from this to this left hand side, so what we have? We will have V0 by Vmax. Okay, and what we will have on this side? We will have S divided by Km plus S. Now in the question it is given that S equal to 9 km. So if you replace this S with 9 km, so what we will have? We will have 9 km divided by Km plus 9 km, and we will have 9 km by 10 km, means the km it cancels out. So we have the answer 9 by 10. So the correct answer is number A, 9 by 10. Just look at the solve a little bit carefully. I will give you a minute to look at it. So now you know how to solve this kind of question. Now any number, if you are given the particular values, you can solve the question easily. Just remember the equation V0 equal to Vmax S by into S by Km plus S. Now if you change the, if you just make it the little bit of rearrangement, so we will have V0 by Vmax equal to S by Km plus S, and now we are replacing the S by 9 km, then we will have V0 by Vmax equal to 9 by 10. It is the answer. So I hope it is clear. If you have any doubt, anybody has any doubt regarding this question, tell me right now if you have any doubt, or it is clear. If it is clear, since we have very many answers for this particular question. Yeah, So, thank you for responding. I will now move forward. So, I hope now if you are given this kind of problem, you can solve it easily. So, now next problem. Problem number 7 says the position where the tRNA will bind with the ribosome depends on it. Number A, codon sequence. Number B, intramolecular hydrogen bonding. Number C, anticodon sequence. And number D, hairpin like structure. So, the position where the tRNA will bind with the ribosome depends on its number A, codon sequence. Number B, intramolecular hydrogen bonding. Number C, anticodon sequence. Number D, hairpin like structure. The position where the tRNA will bind with the ribosome depends on its number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, number D, hairpin like structure. Now, let me check the chat box. Okay, so I have few answers. Anticodon sequence, number C, number C, number A, number B. So, again I have multiple answers, number A, number B, number C. I have 3 different answers for this question. Okay, let us discuss and let us see which answer is the correct one. So, the correct answer will be number C, anticodon sequence. So, this is a structure of a tRNA. We have this 3' portion where the amino, which is the amino acid attachment site. Then we have a loop, a T loop, then we have an anticodon loop and we have a D loop. So, this anticodon, there we have this the particular sequence, okay, 3 sequence of a particular base pair, and based on this sequence are called the anticodon sequence. And this anticodon sequence, why it is called anticodon? Because it is just complementary for this codon sequence on the mRNA. Okay, and the position where the tRNA will bind on ribosome, it is depends on this particular anticodon sequence. So, now how this particular thing, this particular translation process happens, I will just give you a brief about it. So, first what happened, we have this we have a enzyme which is called ribosome, which is important in case of the protein synthesis. Okay, this ribosome has a large subunit and a small subunit. Okay, and then we have this mRNA. On this mRNA we have a start codon which is AUG, and then we have a stop codon, and then we have this tRNA. So, what happens, first the tRNA with the first amino acid, it binds with the first amino acid, and we have an anticodon sequence here and we have the codon sequence here. Now, if you look at the carefully for the sequences, the first we have the anticodon sequence is the UAC. So, now this kind of question can be given to you, like all we know the start codon is AUG, right? The AUG is the start codon and which codes for the amino acid methionine. So, what is the, and first if you are asked what is the first anticodon sequence on tRNA, so remember it is the UAC, which is just the complementary of this AUG. Okay. So, first step of this protein synthesis or protein translation is the initiation stage. What happens here? The translational complex forms and the tRNA brings the first amino acid in the polypeptide chain to bind and to start the codon for mRNA. Okay. So, now this is the translation complex, it is formed first and the tRNA brings the first amino acid to start this particular process. And then we have this elongation process. So, now what is the elongation process? tRNAs bring amino acids one by one. to add to the polypeptide sequence. So, now based on the codon sequence on the mRNA, the tRNA with the particular anticodon sequence brings a particular amino acid, and the particular this chain formation happens, okay. And then now we have this termination process. What happens during this termination process is the release factor recognizes the stop codon, okay, and the translation complex is dissociated and the completed polypeptide chain is released. So, since we have a stop codon, there is a release factor, it will recognize this particular stop codon, and then it will terminate this chain, and we will have a completed polypeptide chain. And then this components are, all the components are recycled, and we have a new cycle for the protein synthesis. So, this is all the very brief about the translational process of the protein synthesis, okay. So, now I will move toward the next problem. So, problem number 8, it says that DNA polymerase enzyme can synthesize number A, DNA into the 5 prime to 3 prime direction, number B, DNA in the 3 prime to 5 prime direction, number C, mRNA in the 5 prime to 3 prime direction, and number D, mRNA in the 3 prime to 5 prime direction. So, how DNA polymerase can synthesize DNA or mRNA? What is the correct answer? We have discussed about DNA polymerase in last to last slide, I guess, and I have told you we will discuss in more detail here. So, from this question we will discuss about the DNA polymerase in more detail. Before that, I want all of you to try to answer the question. DNA polymerase enzyme can synthesize number A, DNA in the 5 prime to 3 prime direction, number B, DNA in the 3 prime to 5 prime direction, number C, mRNA into the 5 prime to 3 prime direction, and number D, mRNA into the 3 prime to 5 prime direction. Let me check the chat box. Number A, number A, number A. So, I think I have number A as the answer, the most given answer. Now, let us check and let us discuss about it. So, the correct answer will be DNA polymerase enzyme can synthesize DNA in the 5 prime to 3 prime direction, which is number A. Very good. So, now let us see the DNA polymerase at the enzyme. It is a group of enzyme basically that can synthesize DNA molecules from deoxyribose nucleotides, the building blocks of the DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single original DNA molecule. During this process, DNA polymerase reads the existing DNA strands to create two new strands that match the existing ones. Also, it performs proofreading and error correction. So, the properties of DNA replications are, DNA replication is semi-conservative, means each DNA strand serves as a template for the synthesis of a new strand, producing two new DNA molecules, each with a new strand and an old strand. This is a semi-conservative replication. Replication begins at an original, usually proceeds bi-directionally. The replication loops always initiate at a unique point, which was termed as the origin. Okay. the one which is the A at T base pair rich sequence. DNA synthesis proceeds in a 5' to 3' direction and is semi discontinuous. A new strand of DNA always synthesized in the 5' to 3' direction with free 3-OH as the point of the DNA is elongated. Okay. So, this is the properties of the DNA replications. So, this is the synthesis of new strand of DNA polymerase with the RNA primer in place. The DNA polymerase catalyzes the synthesis of new strand of DNA into the 5' to 3' direction. So, we have one strand of the DNA which has which goes to the 5' to 3' direction, and for this particular strand the new strand will be forming the particular new strand with the 5' to 3' direction, means which this direction. Okay, the form happens from the 5' to 3' direction. So, remember always DNA polymerase can synthesize the DNA in 5' to 3' direction. So, now I will move toward the next problem. Problem number 9, it says that the type of the topoisomerase which generally relaxes DNA by removing negative supercoiling is number A type 1, number B type 2, number C type 3 and number D type 4. So, which type of topoisomerase generally relaxes the DNA by removing the negative supercoiling? Type 1, type 2, type 3 or type 4? The type of topoisomerase which generally relaxes DNA by removing negative supercoiling is type 1, 2, 3 and 4. Which of the following is the correct answer? I want all of you to respond, and then we will discuss about the topoisomerases which are a very important enzyme during the DNA replication. Somebody wants to join. So, I have few answers. Let others also try. I will give you 1 more minute and then I will show you the answer and then we will discuss. The type of topoisomerase which generally relaxes DNA by removing negative supercoiling is type 1, type 2, type 3 or type 4. Now, let me check the chat box again. Okay, so most of the answers are given for option number A. Okay, let me show you the answer and then let us discuss. So, the correct answer, okay, somebody again was to join, just let me admit. Okay, so it will be number A, type 1. So, supercoiling of DNA, so the topology, the right handed supercoiling is called the negative supercoiling, okay, and the left handed supercoiling is called the positive supercoiling. Then relaxed state has no bends, DNA must be constrained, plasmid DNA or by proteins, unveiling the DNA at one position changes the superhelicity, and topology only defined for the continuous deformation, no strand breakage. Okay. So, the supercoiling of this particular DNA is can be, so how the supercoiling happens. So, basically when there is the formation of single stranded DNA from this double stranded DNA during the DNA replication, I have told you, like the DNA, the first the double helix of the DNA, it breaks into and forms two single strands, and each strand from each strand we have a new double stranded DNA from by the enzyme DNA polymerase. But when you are just unwinding this DNA, there is a tension or the supercoiling at the position of the unwinding. Okay, and this supercoiling or this tension is relieved by this topoisomerase enzymes. So, there are various groups of topoisomerase 1, 2 and so on. So, the difference between the topoisomerase 1 and topoisomerase 2 I will discuss today. So, basically from topoisomerase 1, the enzymes which cut one of the two strands of double stranded DNA relax the strand and reannul the strand. Okay, for topoisomerase 2, these are the enzymes which cut both strands of DNA helix simultaneously in order to manage the DNA tangles and supercoils. The topoisomerase 1 generates single strand breaks and it occurs in eukaryotes, but the topoisomerase 2 generates double strand breaks and occurs in both eukaryotes and prokaryotes. The topoisomerase 1 does not require ATP hydrolysis, and topoisomerase 2 it requires ATP hydrolysis. The subclasses of the topoisomerase 1 is topoisomerase 1A, 1B and 1C, and for the type 2 is type 2A and type 2B. Each and every type has a particular subclass, has particular properties and particular role to play, and which we will discuss slowly. So, the type of topoisomerase which relaxes by DNA by removing negative supercoiling is the topoisomerase 1. Okay. So, now next question. So, problem number 10, it says that the drug that inhibits the initiation step in the translation process is number A atorvastatin, number B nilotinib, number C streptomycin and number D artemisinin. So, which drug inhibits the initiation step of the translation process? Translation process means the protein synthesis process from RNA to mRNA to protein synthesis is called the translation process, okay, which I discussed in last to last slide when I was discussing about the anticodon and tRNA. We discussed about the translation process. So, there was a initiation step. If you remember, there were 3 steps: initiation, and then we have elongation, then we have the termination step. Okay. So, the initiation step is inhibited by a particular drug. So, which one of the following is the correct drug for the inhibition of the initiation step of translation process? Number A atorvastatin, number B nilotinib, number C streptomycin and number D artemisinin. The drug that inhibits the initiation step of the translation process is atorvastatin, nilotinib, streptomycin, artemisinin. Let me check the chat box if you have answers. Okay, I have, I think, multiple answers for this question. Number A, I have streptomycin, number D, number C, number D. So, I think I have number C and number D as the most frequent answer for this question. Okay, let us check what is the correct answer and then we will discuss about it. So, the correct answer will be number C, streptomycin. Okay. So, now let us discuss. So, the mechanism of action for the streptomycin is the streptomycin binds to the 30S ribosomal subunit. I have told you that in the ribosome we have two subunit, one is large subunit and one is smaller subunit. So, 30S is the smaller ribosomal subunit. Okay. So, streptomycin inhibits the S12 and 16S rRNA of 30S small ribosomal subunit encoded by the genes RPSL and RRS. Okay. And the wrong pairing it is responsible for the wrong pairing of codon anticodon reaction and misreading of the genetic code, and this blocks the translation. So, this is the for the resistant, sometimes the resistance happens of this particular streptomycin because of the point mutation of this particular two genes RPSN and RRS genes. So, how the protein synthesis inhibition happens? So, there are various drugs, different drugs which inhibits this particular protein synthesis. Okay. So, we have this is particularly your large ribosomal subunit which is 50S portion, and this is the smaller ribosomal subunit which is 30S portion. This is the messenger RNA or mRNA, and this is the tRNA. So, tRNA we have the anticodon sequence here, and on the mRNA we have codon sequences here. So, streptomycin, what does the streptomycin do? So, basically the streptomycin changes the shape of the 30S portion, okay, and it causing the code of the mRNA to be read incorrectly. So, since this particular streptomycin is bending this particular mRNA, and that is how this tRNA cannot read the particular codon with this anticodon sequence, and it blocks this particular protein synthesis. Okay. And it is the initiation step. Okay. Initiation step is when the codon anticodon interaction happens, and this is inhibited by this particular streptomycin. And then we have this the various different kind of drugs which are responsible for this stopping of this translation process. We have this chloramphenicol, it binds to the 50S portion and inhibits the formation of peptide bond. Okay. And for this there is a one amino acid, there is a one amino acid. So, if this is a protein synthesis site, and the chloramphenicol it binds to this 50S subunit, and then if it is here there will be no peptide bond formation between two amino acids, and the elongation step is inhibited. And we have tetracyclines, they interfere with the attachment of tRNA to mRNA ribosome complex, and this is also inhibits the initiation step for this particular protein synthesis. So, there are various drug, and if you are wondering why we need to inhibit this particular protein synthesis, so basically these drugs are used as a various agents, like mainly these drugs are used as antibacterial agents, because they inhibit the protein synthesis in particular bacteria, and that is how they exert their antibacterial activity. Okay. So, now I will move toward the next problem. Problem number 11 says, which is of the following is the start codon and for which amino acid it is code for. I think I have discussed it in many times, but still I want all of you to answer this question. Number A, AUG and methionine. Number B, UCU and serine. Number C, UUU and phenylalanine. And number D, AAA and lysine. I know all of you can answer this question correctly, so just write which one of the following options are correct, and then we can discuss and move forward. Which of the following is the start codon and for which amino acid is code for? I already have discussed it many times in previous slides. Let me check the chat box. I know all of you can answer this question very correctly. Yeah. So, it is number A. Very good. So, the start codon is AUG and the amino acid for which it codes for is the methionine. So, transcription, as I have mentioned, that it is the first step of the protein synthesis is the transcription. What is the transcription? It is the formation of the mRNA in the nucleus by complementary base pairing of the DNA. mRNA then leaves the nucleus and travels to the ribosome. Ribosome read the mRNA code, and codon is the sequence of the 3 base on the mRNA. So, there are 64 different codons and 20 different amino acids. Several amino acids have several different codons that code for them, and one codon is the start codon, which is AUG, and which codes for methionine. In case of eukaryotes, in case of prokaryote, it is the N-formyl methionine. Okay. So, now next question. So, problem number 12 says fetal hemoglobin has a higher binding activity for the oxygen than does the adult hemoglobin. In comparison to the adult oxyhemoglobin dissociation curve, the fetal hemoglobin oxyhemoglobin dissociation curve will be: number A, shifted to the left displayed and display lower Km, number B, shifted to the right and display lower Km, number C, shifted to the right and display higher Km, and number D, shifted to the left and display higher Km. Fetal hemoglobin has a higher binding activity for the oxygen than does the adult hemoglobin. In comparison to the adult oxyhemoglobin dissociation curve, somebody wants to join. So, I will repeat the question number 12. The fetal hemoglobin has a higher binding activity for oxygen than does the adult hemoglobin. In comparison to the adult oxyhemoglobin dissociation curve, the fetal oxyhemoglobin dissociation curve will be: number A, shifted to the left and display lower Km, number B, shifted to the right and display lower Km, number C, shifted to the right and display higher Km, and number D, shifted to the left and display higher Km. Read the options carefully, read the question carefully, and then answer. This type of questions can be little tricky. Whenever there is a curve and there is a graph kind of questions, they can be little bit tricky. So, read it carefully and then answer it. Let me check the chat box and then we can discuss. Okay. So, option A is, I think, most given answer. Okay, and few of you have answered it. Okay, let us discuss. So, the correct answer will be, yes, option A. It will shift to left and display lower Km. So, now let us discuss what is the first. We will discuss what are the differences between the adult hemoglobin and the fetal hemoglobin. So, adult hemoglobin, it has 2 beta chain and 2 alpha chain. Okay, but for the fetal hemoglobin, which is adult hemoglobin is abbreviated as HbA and the fetal hemoglobin as HbF. So, in case of fetal hemoglobin, we have 2 alpha subunit and 2 gamma chains. Okay, the polypeptide gamma chains enable the fetus to have greater oxygen affinity toward the oxygen. It can ensure enough that ensure that enough oxygen is being delivered to the organs of the developing fetus. Okay. Now, what are the differences? So, fetal hemoglobin has a higher affinity toward the oxygen at all partial pressures. So, if you can see the graph carefully, this is the percentage of the saturation of the hemoglobin with oxygen. Okay, and this is the partial pressure of oxygen. So, this is the F curve for the adult hemoglobin and this is the curve for the fetal hemoglobin. the fetal hemoglobin has a higher affinity of oxygen at all partial pressures of oxygen. So at all partial pressures it have a higher affinity of the oxygen. So this ensures that oxygen is transferred to the fetus from the maternal blood across the placenta. The pressure of oxygen in the fetal tissues is very low due to the high metabolic rate associated with the fetal growth rate. Okay, although the fetal hemoglobin has a higher affinity toward the oxygen in a such low partial pressure environment of the fetal tissues, it unloads the readily. Okay, at the birth the fetal hemoglobin is replaced by adult type of hemoglobin. So as you can see, this particular for the curve for the fetal hemoglobin it is shifted toward the left side. Okay, since it has a higher affinity to oxygen to high at all partial pressure, and it displays a lower Km. So how it displays a lower Km? So we all know Km is the particular it is similar as your Michaelis-Menten, right? So the Km is the at a 50% affinity, the it is the partial pressure of, if you see the 50% affinity, and the Km is the Km will on be the, if you draw a perpendicular, then we will have a Km. So it is not a perpendicular, just let me erase it a little bit. So if you draw a perpendicular from this curve on the x axis, so you will see, okay, this is not perpendicular, yeah. So this is the Km for adult hemoglobin, this is the Km for fetal hemoglobin. So as you can see, the fetal hemoglobin has a lower Km, and the curve is shifted toward the your left side. Okay, so remember these things, how the curve look like for the adult hemoglobin and the fetal hemoglobin. And also remember that for in case of adult hemoglobin we have 2 alpha and 2 beta chains, and for case of fetal hemoglobin we have 2 alpha and 2 gamma chains, and due to these 2 gamma chains we have higher affinity toward the oxygen for the fetal hemoglobin, which is important for the developing fetus. Okay, so now I will move toward the next problem. So problem number 13 says, where does the inhibitor binds on enzyme in mixed inhibition? Number A, allosteric site. Number B, active site. Number C, it binds on the substrate. Number D, it does not bind on the enzyme. Where does the inhibitor binds on the enzyme in case of mixed inhibition? Allosteric site, active site. It binds on substrate. It does not bind on enzyme. Which one the following will be the correct answer? All of you try to answer the question. As I have said, even if your answer is wrong, it doesn't matter, but I want all of you to participate. After you try to answer this question, then I will show you the answer and we can discuss it. What is mixed inhibition and what where the inhibitor binds during the mixed inhibition, we can have a full discussion on it. But before that, I want all of you to try to answer this question. Your options are number A, allosteric site, number B, active site, number C, it binds on substrate, and number D, does not bind on the enzyme. Where does the inhibitor binds on the enzyme in mixed inhibition? wants to join. Let me check the chat box and then we can answer. Okay, yeah, lower Km means higher substrate concentration, that is for oxygen, and it will be shift to it. Yeah, it is correct. So number A, number A, number D, number A. So I have number A or number D for the answer of the problem number 13. Now let us discuss and let us check first what is the correct answer, number A or number D, and then we can discuss. So the correct answer, where does the inhibitor binds on the enzyme in mixed inhibition, will be number A, allosteric site. So first let us see what is the difference between the allosteric site and active site. So an allosteric site is a region on an enzyme where activator or inhibitor molecule can bind and either activate or decrease the enzyme activity. This is only found in allosteric enzymes, and it is also it can have substrate and do not have two separate websites. The active site of enzyme is the area where the substrate molecules bind and the enzyme catalyzes the reaction, where the creation of specific products happen. All enzymes contain it, and effectors molecules can also bind to the active site, and binding and catalytic sites are two different sites. Okay, in case of the particular active site, binding site, catalytic site are two different sites. So in non-competitive inhibition, there is a particular your enzyme, and we have the substrate binds on the active site, and there is a inhibitor binds to a allosteric site. And in case of enzyme inhibitor complex, we have a different active site, the active site changes, and since inhibitor is binding to the allosteric site, there is a change the shape and size of the active site, and now we do not have any kind of interaction between the substrate and the enzyme. So in case of mixed inhibition and non-competitive inhibitions are the same. Okay, so in case of mixed inhibition or non-competitive inhibition, what happens that there is a allosteric site and there is a active site. The inhibitor molecule first binds to the allosteric site. Okay, and if inhibitor molecules bind to the allosteric site, so now the particular your substrate, it cannot bind to the particular active site, and we do not have any product formation, enzyme substrate complex formation, and we do not have any product formation. So the inhibitor molecule binds to the allosteric site for in case of mixed inhibition or non-competitive inhibition. So now we will move toward the next problem. Problem number 14 says a student is conducting an experiment in which he adds an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return the reaction rate to the normal by adding up a large quantity of substrate. What type of inhibitor is he using? Again, I will repeating the question. A student is conducting an experiment in which he adds up an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return the reaction rate to normal by adding up a large quantity of substrate. What type of inhibitor is he using? Your options are number A, non-competitive inhibitor, number B, a competitive inhibitor, number C, an uncompetitive inhibitor, number D, the student likely made a mistake, the result is unacceptable regardless of any inhibition. So read this question very carefully, think a bit and then answer this question. Which kind of inhibitor the student is using? A student who is conducting an experiment, he adds an inhibitor to an enzyme catalyzed reaction, and first after adding up the inhibitor, the reaction rate is decreasing, but he can return the reaction rate to normal by adding up large quantity of substrate. So now what type of inhibitor is he using? Non-competitive, competitive, uncompetitive, or he made a mistake, the result is unacceptable. Read the question, read the options very carefully, then write your answer. Let me check the chat box and then we can discuss. Number B, number A, number B. So I have most number A and number B as answers. Let us check which. So the correct answer is number B, competitive inhibitor. So see, in case of competitive inhibitors, what happens? There is an enzyme and we have a substrate, but the competitive inhibitor, what does it do? It binds to the active site of the particular enzyme where the substrate binds. Generally the substrate binds to the active site, but the competitive inhibitor, it also binds to the particular active site of the enzyme, and if the inhibitor is binding to the particular active site, now the substrate cannot bind to the active site and no product formation happens. So this is a competitive inhibition where the inhibitor molecule binds to the active site, and in case of mixed inhibition and non-competitive inhibition, the inhibitor molecule binds to the your allosteric site, right? And so this is the Michaelis-Menten equation and Michaelis-Menten curve for this particular competitive inhibitor. Okay, so the blue one, the bluish one, and we have this reddish one. Okay, this is the normal one and this is the inhibition one. Okay, so particularly what happens generally when the student is adding the inhibitor, the enzyme catalyzed reaction. So now what is happening when there is a competition between the particular the inhibitor molecule and the substrate molecule for binding to the particular active site of the enzyme. But if he increases the amount of the substrate, so now what happens if the amount of the concentration of the substrate is higher, so there is a particular more chance of the substrate to bind to the active site, not the inhibitor molecule. That is why when there is a large quantity of the substrate, the rate becomes normal. Okay. So this is the competitive inhibition basically. So there are one example of the competitive particular inhibitor is this succinic dehydrogenase enzyme and the succinate and the malonate. Okay. So succinate is the particular substrate for the succinate dehydrogenase enzyme, but if you use malonate, malonate acts as an inhibitor molecule, and malonate has a similar structure like succinate, and if it is bind to the this active site of the succinate dehydrogenase, no fumarate formation happens, which is a normal reaction, and this is a particular curve. Okay. And as you can see from this curve, okay, this is one type of particular enzyme inhibition curve. This is also, and this is the Michaelis-Menten curve, and if you can see this curve, this 1 by V by 1 by S curve. So you can see the 1 by V max is same for this two portions. So which implies that at a higher velocity or higher substrate concentration, both the reaction rate will be equal in case of competitive inhibitor. Okay. So now next problem. Problem number 15 says the Lineweaver-Burke plot is associated with number A, straight line with the slope of minus km by V max, number B, parabola with the intercept of 1 by V max, number C, straight line with the slope of km by V max, and number D, parabola with the intercept of minus 1 by V max. So Lineweaver-Burke plot is associated with which one? So this plot is similar to the Michaelis-Menten plot. This is a one kind of enzyme kinetics plot. Just we have this Michaelis-Menten curve. Lineweaver-Burke plot is another one. So now you have to identify what is the correct statement, which is what is the with which option this is this plot is associated. Number A, straight line with a slope of minus km by V max. Number B, parabola with the intercept of 1 by V max. Number C, straight line with a slope of km by V max, and number D, parabola with the intercept with minus 1 by V max. Let me check the chat box if there are any answers for this question. Number A, number C, number A. So I have number A and number C. Okay, let us check. So the correct option will be number C, straight line with a slope of Km by Vmax. So this is the Lineweaver-Burke plot. Okay. So as you can see, on the y axis we have 1 by V and 1 by, on the x axis we have 1 by S. Okay. And this slope of this particular straight line. So if we plot this graph, so we have a particular straight line where we have this slope of Km by Vmax and the intercept of 1 by Vmax. Okay. And so sometimes there we know there are different kind of equation for this enzyme kinetics. We have this Michaelis-Menten equation, which is V0 V equal to Vmax S into Km divided by Km plus S. We have Michaelis-Menten constant where Km is K2 plus K3 by Km. We have Lineweaver-Burke equation, which is 1 by V equal to Km by Vmax into 1 by S plus 1 by Vmax, and Hanoi-Wilf equation can be derived from this equation. So all these equations, sometimes students they get confused between all this kind of equations. They think that how can we remember so many equations, but let me tell you, only equation you have to remember is this Michaelis-Menten equation. It is also can be derived actually. So which we can, I think, discuss more detail in the last summary classes. So this can, if you remember this equation, so you can derive this Lineweaver-Burke equation and this Hanoi-Wilf equation from this Michaelis-Menten equation itself. Let me show you how. So we have this Michaelis-Menten equation, V equal to your Vmax, I am writing it as Vm into S divided by, okay, we have this Km plus your S. So this is your Michaelis-Menten equation, right? Now if just one thing you have to do is, we have to take the reciprocal of this whole equation, just you have to make it 1 by V. So if you make it 1 by V, so what will happen? Now we have this Km plus S as the numerator, and as the denominator we have this Vmax into S. Now if you just split this particular thing, so what we will have? We will have 1 by V equal to Km by Vmax or Vm into your 1 by S, right? And then we have plus 1 by Vmax. So this is your Lineweaver-Burke plot. If you look at this, so nothing you have to do, you have to just take the reciprocal of this Michaelis-Menten equation, then you will derive this Lineweaver-Burke equation. Now from this equation, having this Hanoi-Wilf equation is again easy. You just have to multiply this whole Lineweaver-Burke equation with this concentration or substrate concentration of S, then we will have this Hanoi-Wilf equation. Now this is an equation of straight line, right? If you compare it with y equal to mx plus c, if you compare y equal to mx plus c, so you can see this m is the slope, which is Km by Vmax, and your c is the intercept, which is 1 by Vmax. Okay. So this is the particular equation of Lineweaver plot. So it is very easy. If you remember Michaelis-Menten equation itself, then you can easily convert it to the Lineweaver-Burke equation by taking the reciprocal, and you can convert Lineweaver-Burke equation to the Hanoi-Wilf equation by just multiplying it by substrate concentration. Okay. So now next question, problem number 16 says, which inhibition study deals with the decrease of the value of both Vmax and Km with the increase of the inhibitor concentration? Your options are number A competitive, number B non-competitive, number C uncompetitive, and number D suicide inhibition. Which inhibition study deals with the decrease in the value of both Vmax and Km with increase in the inhibitor concentration? Number A competitive, number B non-competitive, number C uncompetitive, number D suicide. Which deals with the decrease of both the values of Vmax and Km with increasing the inhibitor concentration, and we have to consider the Lineweaver-Burke plots of the particular inhibitions in case of this particular question. Take a time, think a bit and then answer the question. Which inhibition study deals with the decrease in the value of both Vmax and Km with the increase of the inhibitor concentration? Let me check the chat box. Okay, so I think I have few answers. Number D. The responses are getting decreased. See, since there are no marks, I want all of you to try, okay. Even if your answer is wrong, you should try to answer these questions. So, let me check. So, the correct answer will be, it is number C, uncompetitive inhibitor. In case of uncompetitive inhibitor, the decrease with the value, both with the value of Vmax and Km is decreased with the increase of the inhibitor concentration. So, now these are the Lineweaver-Burke plots for the particular enzyme inhibition. The first one is in case of competitive, now the second one is uncompetitive and the third one is the non-competitive. So, in case of the competitive inhibition, as you can see, the red one is for the inhibitor and the blue one for the non-inhibitor. Okay. So, as you can see in both of the cases, these lines are intersecting on the y axis on a same point, okay. Means the interceptor, the value of intercept is same. The value of intercept means 1 by Vmax. If 1 by Vmax is same, means Vmax is unaffected, but if the Vmax is, since the Vmax is unaffected, since the slope is increasing, means the Km is increased. The slope is Km by Vmax. And if the, as you can see with the inhibitor, the slope is increasing. This red one is the much steeper one. Means with inhibitor the slope is increasing, means the Km, with increasing the substrate concentration, we have what we have this, sorry, with increase of inhibitor concentration, we have this Km increased, but the Vmax is unaffected. But in case of the uncompetitive inhibitor, as you can see, the both of the slopes are the same. The slopes with inhibitor or without inhibitor, the slopes of the are the same, but there is a difference in the Vmax. Okay, as you can see, the value of intercept is higher. Okay, the value of intercept is higher in case of with in presence of inhibitor. The value of intercept is higher means the value of 1 by Vmax is higher. If the value of 1 by Vmax is higher, means the Vmax is getting reduced, and since the slope is same, means the Km is getting reduced with the same manner in case of this particular uncompetitive inhibition. But in case of non-competitive inhibitor, what happens, the Vmax is reduced again, okay, but the Km remains unaffected. Since the Km remains unaffected and the Vmax is getting reduced, so how the slope changes? If this is the Km is unaffected and Vmax is reduced, means the slope increases. Okay. So, that is how this particular inhibitions happen. Okay. So, here we have the question that both Vmax and Km is getting reduced in case of uncompetitive. In case of the competitive inhibition, Km is increased and Vmax remains unaffected, and in case of non-competitive, Km is getting remaining unaffected and Vmax is getting reduced. Okay. So, just remember how this particular different kind of Lineweaver-Burke plots are looks like for in case of different inhibitor, and if you remember these things, you can easily answer this kind of questions. Okay. So, now I will move forward the next problem. So, this is a mathematical problem. I want all of you to take some time, but answer this question. Okay. Consider the following plot of the velocity versus substrate concentration of the enzyme catalyzed reaction obeying Michaelis-Menten kinetics with an initial enzyme concentration of 10 to the power minus 5 micromolar. The turnover number for this reaction is 8,000, 8 lakh, 40,000 and 4 lakhs. This is the particular graph. Okay. You have to consider this graph and then you have to calculate the turnover number for the reaction. Take some time, I will give you some time and then try to solve this problem. Next we have few mathematical problems now on. So, I want all of you to try to solve the questions. I will show you how to solve it in detail, but before that I want all of you to try. All of you try to answer, try to solve this kind of problem. We will have few mathematical problem for next 2 to 3 question, I think. Just you have to try a little bit because these questions are very easy. Let me check if we have any answer in the chat box for this question. Okay, I have few answers, number D, number D, number, okay, I have only number D as answer. Others? Okay, since we have not much time left, so let me show you what is the correct answer and then let us see how to solve this kind of question. So, the correct answer will be number B. Okay. So, how to solve this kind of problem? So, this is the Michaelis-Menten equation. Okay. So, Michaelis-Menten equation we always, so the most detailed form of Michaelis-Menten equation is this one. Okay. V0. So, we all know the Michaelis-Menten equation which we are used to looking at is V equal to V max into the substrate concentration, okay, divided by the Km plus substrate concentration, right. But this particular V max, it can be written as K2 into E0. So, what is K2 and what is E0? So, basically K2 is known as the E0 is the enzyme concentration, okay, as this which is the enzyme total enzyme concentration is known as the E0. So, the main Michaelis-Menten equation can be written if we just write K2 into S0 in case of the position of Vmax. So, we have this particular equation, okay. So, at higher substrate concentration at the high, so this is the overall equation, okay. So, at high substrate concentration means the highest concentration, we have this S0 much greater than Km. So, when the substrate concentration is much greater than Km, so we can neglect this Km in the we can neglect this Km, and if we neglect this Km, so what we have rate equal to K2 into E0 equal to Vmax, okay. So, that is the rate attains the maximum value and the reaction is of 0 order with respect to the substrate concentration. This K2 is known as the turnover number for the enzyme, and it represents the number of molecule that is converted to product in unit time a unit by 1 molecule of enzyme. This is very important. What is the turnover number of the enzyme? So, remember what is the turnover number? Turnover number, represents the number of the molecule converted to product at unit time by one molecule of enzyme. So, now according to question, what the question is given, according in the question it is given that the initial enzyme concentration is 10 to the power minus 5. Initial enzyme concentration means E0. E0 is 10 to the power minus 5, and the Vmax, if we just look at the plot carefully, so you will see, if you extrapolate this portion, this plateau, you will have this Vmax in micromole per minute, we have this 8. Okay. So, Vmax equal to 8 micromolar per minute. So, K2 will be Vmax by 2, as we know the Vmax equal to K2 into E0. So, now K2 will be Vmax by E0, which is 8 micromolar into 10 to the power minus 5 divided by 10 to the power minus 5 minute inverse, which is this 8 lakh minute inverse. So, this is the correct answer. Okay. When I share this PPT, just look at this problem very carefully and look at the answer. I think you will easily solve this kind of problem if it is given to you. Just remember what is the turnover number and how to solve this kind of question. So, now you can solve any kind of this kind of question. Okay. So, now next problem. Problem number 18 says, consider the following plot of the 1 by V0 versus 1 by is for the Lineweaver bark. Okay, I just forget to give you the picture, but it is the normal Lineweaver bark plot. Okay, the result obtained from the Lineweaver bark plot is the intercept value is 0.1 micromolar minute inverse and the slope is 9 into 10 to the power 5 minute. What is the Km of the reaction? Your options is number 1 9 into 10 to the power 5 micromolar, number B is 0.11 into 10 to the power 5 molar, number C 9 into 10 to the power 6 micromolar, number is 0.011 micromolar. Lineweaver bark plot we have discussed so far many times, and I have told you already what is the intercept and what is the slope of Lineweaver bark plot. Now, just you have to find out what is the Km. The values are given. So, I know all of you can solve this question. Just take 2 minutes, 2 to 3 minutes and try to solve it, and let me know what is the correct answer in the chat box. I will give you 2 minutes. Okay, let me check the chat box if there are any answers, and then we can discuss it, and I can show you how to solve this kind of problem. These are very easy actually. Okay, I think I have one answer, which is number C. Okay, let me check if number C is the correct answer. So, the correct answer will be, very good, number C. So, how to solve? So, Lineweaver bark plot, I have mentioned Lineweaver bark plot earlier, right? We have discussed it. Here it is. So, this is the Lineweaver bark plot. If you remember, we have 1 by V on the y axis and 1 by S on the x axis, and I have told you the value of your slope will be Km by Vmax and the intercept is 1 by Vmax. Now, back to our problem. It is already has given all the values. So, this is the equation of Lineweaver bark plot. 1 by V0 equal to 1 by Vmax plus Km by Vmax into 1 by S. Now, if we plot 1 by V0 versus 1 by S, so we will have this following information: the intercept is 1 by Vmax and the slope is Km by Vmax, right? So now from the equation it is given that 1 by Vmax equal, or the intercept value is 0.1 micromolar per minute inverse, and Vmax equal, so the Vmax will be 10 micromolar per minute, and the slope is Km by Vmax. So now you have to find out the Km. So Km will be slope into Vmax, which is 9 into 10 to the power 5 minute into 10 micromolar per minute, which is 9 into 10 to the power 6 micromolar. So number C is the correct answer. So now I will move toward the next problem. Problem number 19 say, consider the plot of S by V0 and S plot of the Hanoi wolf. The results obtained from the graph is, the intercept is value is 0.5 and the slope is 0.0002 millimolar inverse. What is the Km of the reaction? Again I have told you the difference between Hanoi wolf and the particular Lineweaver bark plot is very easy. Just you have to multiply the whole equation with the substrate concentration. Now just write Lineweaver bark plot equation, multiply it with S, then we will have Hanoi wolf equation. From that you will easily have the intercept and slope value, and if you put the given among the quantities, you will have this particular Km. Yes, somebody wants to join. This is the Lineweaver bark plot. You just have to multiply the whole equation with concentration of S, then you will have the Hanoi wolf equation, and if you put the values you will have this particular Km value, because the intercept and slopes are given. Just try for some time and then I will show you. Let me check the chat box, then we can solve it. Okay, let me show you how to solve this kind of question. Whenever you are reading about the enzyme kinetics, just not only Michaelis-Menten equation, read about these two or other equations also, because question can come from these kind of these equations, because they are not very much common but they are very easy also. So the correct answer is number C. Now I will show you how to solve. So the Hanoi wolf equation is this: substrate concentration by V0, S by V0 equal to S by Vmax plus Km by Vmax. So here it is Y equal to Mx plus C if you compare it with it. So we have intercept value Km by Vmax and we have slope 1 by Vmax. So now from the question it is given the Km by Vmax is 0.5 and 1 by Vmax is 0.0002. So now Vmax equal to, if you just calculate all this thing, we have this 2500 millimolar. Okay. So now last question of today's session, it is, which of the following agent does not have any effect of its own? The options are number A agonist, number B antagonist, number C partial agonist, and number D inverse agonist. So which of the following agent does not have any effects of its own? Agonist, antagonist, partial agonist, or inverse agonist? This is the last question of today's session. I want all of you to answer. which of the following agent does not have any effect of its own? Agonist, antagonist, partial agonist, inverse agonist. Let me check the chat box. Okay, number D, number C, number D. I have few answers. Let us check. So, the correct answer will be number B, antagonist. So, a macromolecule or so the drug receptor interaction, there are few terms which are used for drug receptor interactions like agonist. Agonist is an agent which activates a receptor to produce an effective similar to that of physiological signal molecule. Okay, all these things are related with the receptors, okay, the drug receptors. Okay, so the drug receptors, what is a drug receptor? It is a macromolecule or a binding site located on the surface or inside the effector cell that serves to recognize the signal molecule of a drug and initiate the response to it, but it does not have a function of its own. Okay, so there are different agents which are used to describe the drug receptor interaction like agonist. Like agonist is an agent which activates the receptor to produce an effect similar to that of the physiological signal molecule. Inverse agonist is an agent which activates the receptor to produce an effect opposite to the direction of the agonist. Antagonist is an agent which prevents the action of agonist on the receptor on subsequent response, but it does not have any effect of its own. Partial agonist, an agent which activates the receptor to produce submaximal effect, but antagonizes the action of full agonist. A ligand, any molecule which attached selectively to particular receptors on sites. The term indicates the affinity or ability to bind without the functional change. Agonist and competitive antagonist are both ligands of the same receptor. So, antagonist is the agent which does not have any effect of its own, but it prevents the action of the agonist on a receptor. Okay. So, this was the last question of today's session. So, what did we learn today? We learned about many things of enzyme inhibition. We learned about many various kind of inhibitors molecule. We learned about competitive, non-competitive, uncompetitive inhibitors, and we solved many mathematical questions also. Although there may be the mathematical questions may seem to be little bit difficult for the first go, but when I will share you this PPT to you, and if you practice 1 or 2 times, I think you will find it very easy to solve this kind of mathematical questions. Then we discussed about the nucleic acids, the DNA, the RNA, we discussed about DNA replication, we discussed about protein synthesis, many things during today's session. So, now if you have any doubt, any query, or you want me to repeat anything, you can let me know in the chat box. We have 1 or 2 minutes I think left in today's class. So, if you want me to repeat anything, you can tell me. I am sharing my email ID with you here, okay, in the chat box. So, if you have any doubt or any query, so this is my email ID. So, if you want anything, you want to tell me about the session, about the questions, about the topic, anything, you just feel free to drop an email to me. I will happy to share your queries. So, today we had our week 2 live session. Every week we will have this kind of live session on Tuesdays from 6 to 8 p.m. I want all of you to join on time so that we can discuss as much as we can, and any doubt, any query, just feel free to ask me. Okay. This lecture PPT and this video, the class recording will be shared to you on the SWAM portal. So, feel free to refer to this particular things. So, thank you so much all of you to join today's class. All of you have responded very well in today's class, and that is very important. Now, let us meet on next Tuesday from this time 6 to 8 p.m. and if you have any problem, feel free to contact me on this email ID, or you can also write on the NPTEL forum. So, I will stop today's session and thank you all again for joining. We will meet on the next class. So, now I will close the session for today. Thank you all.